The next category of antibiotics we want to talk about are protein synthesis inhibitors. So recall that you have 80 S ribosomes in your eukaryotic cells. Bacteria have 70 S ribosomes in their cells. And the fact is that there's enough differences between those that there are whole sets of drugs that target differences between them. Many of these drugs either bind to the 50 S ribosomal subunit or the 30 S ribosomal subunit in prokaryotes and interfere with various ways that proteins get synthesized. I will make a comment that, you do have mitochondria, and we know that they came to be in your eukaryotic cells via endosymbiotic theory, and they can be affected by high levels of some of these drugs as a result. Usually it's not a problem, but we do tend to think of these as having a lot more potential toxicity than the cell wall inhibitors, for example. So I went too far here. So these are some of the major classes of protein synthesis inhibitors. So here's just a reminder of where we are. Here's a ribosome. Here's our large 50S subunit, our small subunit, a 30S. And here's our messenger RNA that's being translated, right? And here we go. So here's our growing polypeptide chain. So the arrows sort of point to the major areas where we see these drugs affect protein synthesis. So the chloramphenicol, macrolides, lincosamides, they bind to the 50S ribosomal subunit, and they prevent peptide bond formation, so they're going to shut down protein synthesis. Aminoglycosides bind to the 30S ribosomal subunit. They impair proofreading. So sometimes the cell can make proteins, but the proteins are all messed up, so they don't work. So those is also really effective. And then tetracyclines, which you've probably already heard of, bind to the 30S ribosomal subunit, but they block the binding of transfer RNAs. So when that happens, you can't get your new amino acids brought to the ribosome, and you can't synthesize protein. So all of these are different ways that these protein synthesis inhibitors work. Some just examples of 50S subunit interferers. Chloramphenicol is a really good example of those. They're broad spectrum. All bacteria make proteins. All bacteria have 70S ribosomes, so it makes sense that these would be broad spectrum. Another 50S subunit interferer is clindamycin. Sometimes people are familiar with this because it's used so commonly to treat acne. So this is Cleocin T is the brand name of clindamycin, and this is a little pad, and it kind of absorbs the clindamycin. You kind of pat it on your skin. Generally, this works for a couple of years, and then your bacteria develop resistance and it stops working, but it's nice while it lasts. This is actually, this and other protein synthesis inhibitors tend to have significant adverse GI side effects. This one in particular, which is why we use it topically, but in general, these tend to have more sort of stomach upset associated with them than other antibiotics. Here's a 30S subunit interferer, so really famous tetracycline. I'm sure you've heard of it before. It prevents the attachment of transfer RNA, so the ribosome can't add amino acids to the growing peptide chain. They do some good things. So tetracyclines are used to suppress the intestinal bacteria superinfections. So when a bacteria that shouldn't be there is really taking over your intestinal flora, this can help with those. But it has some bad side effects. It can cause liver damage in pregnant women. And one of the side effects that's really well understood is that it can cause tooth discoloration in children. So this is a picture of someone whose parent was either exposed to tetracycline or they were exposed as a young child. And what happens with tetracycline is if the tooth that's developing is exposed to tetracycline during the period of time when it would be undergoing mineralization or calcification, the tetracycline actually binds to calcium ions in the teeth. And then when the teeth erupt through the gums, the tetracycline initially kind of looks sort of like a fluorescent yellow, but when the light hits it, the tetracycline is oxidized and it turns into this sort of brown period over a period of months to years. So where the brown teeth are correlates to when the person was exposed to tetracycline. Usually you see a little bit of a less intense discoloration in permanent teeth, than in primary teeth, but they can discolor both kinds. So when women are pregnant, we don't give them tetracycline unless, you know, they're going to potentially die if they don't have it because it causes this particular effect. All right, so the last set of slides that I'm going to go through are the other mechanisms. So the two big ones are protein synthesis inhibitors and cell wall inhibitors, but we're going to look at the other three mechanisms as a group.